So thank you everyone again for joining us. Today I'm going to be sharing some information about upcoming events and then we'll hear from our featured speakers and we can go from there. Like I said, please feel free to use the chat and question and answer throughout this session. We'll be monitoring those as we go. Just a reminder that if you have not been able to catch up on our previous webinars, is available on our YouTube. We moved some of the videos over to a new channel just so everything is organized, but there's a playlist specifically for CFDE. So I just wanted to alert everyone to that change as of now. And with that, just a reminder, if you have any questions, feedback, or would like to be added to our listserv, please feel free to email us at help@cfdeknowledge.org. So with that, we have some various conferences that we'll be attending as CFDE, and coming up next week is the BioIT World Conference in Boston, which I'll talk a little bit more about in a minute. In July, the CFDE will be at the Intelligent Systems for Molecular Biology conference in Liverpool. We have some exciting events coming up for that, and I know there are various members of the CFDE who will be attending as well. So hopefully we can plan some time to connect with each other for all of those that will be at this event. And in October, we'll be at ASHG in Boston, and we're planning to have both a booth and ancillary session again this year as we had last year. This detailed information will come out a little bit closer to the events as we are still in the planning stage for all of these, but please mark your calendars. And so, as I mentioned, BioIT World is coming up. So if you will be attending, the CFDE Training Center will be hosting the 6th annual BioIT Fair Data Hackathon, which will be April 1st and 2nd. This session specifically will allow attendees to collaborate with data scientists, developers, and life science professionals to tackle real-world data challenges. Just a reminder that this event is free, but you must register to participate. This is the link here, and we can share that with everyone as well. There are some various projects that you can participate, and if you have any questions, please reach out to us. Another hackathon that you can attend will be hosted by Bridge to AI Voice, and this will be held in Tampa, Florida. Attendees basically can learn about AI and machine learning in voice biomarker health research. So keep an eye out for that as well, as that's coming in April. One other thing that I wanted to highlight specifically is the CFDE has an exciting new podcast hosted by Dr. Alyssa Dillman called Decoding the Data Ecosystem. Now, this is just more than a podcast. It's actually built as a community for those who are passionate about potential omics of researches to solve complex biological puzzles and addressing pressing health challenges. So this is for anyone to participate in, whether you're someone who is deeply skilled in research or you're just starting out as a trainee. This podcast is basically able to give you insight, stories, and advice as you're traversing through omics training and education. So with that being said, there are 4 episodes that are currently out and have featured speakers who represent various programs and initiatives within the ecosystem. This is all available on the Blueberry site, which was linked on there. We can also share that as well. And we highly encourage you to take some time to dedicate to listening to this session and learning more about the research that's going on within the ecosystem. And so with that, I would like to introduce Julie, who will be presenting on behalf of the Knowledge Center. Today, she'll be discussing some production release updates, which include the PIGEAN method and a new beta resource connecting mouse to human phenotypes. So as a reminder, please feel free to utilize both the chat and question and answer. I will be monitoring that as we go, and we really would like to have an interactive session with you today. So with that, Julie, I will hand this over to you.
All right. Thank you so much, Mackenzie. I'm going to share my screen real quick. And there we go. So hi, everyone. For those who don't know me, I'm Julie Jurgens. I'm a representative of the CFDE Knowledge Center. I'm going to talk today about how you can obtain knowledge through the Common Fund Data Ecosystem, or CFDE, and ways of getting open access program information, analysis results, and more. So for those who are new to the ecosystem, we're going to start out somewhat generally, give you a little bit of a background about what CFDE actually is. And then we'll go into some more specifics and some updates from the Knowledge Center on new tools that you might be able to apply to your research. So many of you are probably familiar with the NIH Common Fund. As the name suggests, this is a funding mechanism, and it supports multi-center, multi-institutional collaborative research. The major goal of the Common Fund is to catalyze discovery and to address sort of big picture challenges that face multiple research branches. So not just a single NIH department or division. The goal of the Common Fund is to significantly change the trajectory of biomedical research. And Common Fund program support is designed to be time limited. So major themes that you see across Common Fund programs are accelerating emerging science, removing roadblocks that come up to research, building up the research workforce, and finally supporting high risk and high reward science. Common Fund programs themselves are designed to support these three sort of major arms of research. So they can transform science and discovery, catalyze data resources, and build these sort of repositories for scientific discovery, and finally re-engineer the research workforce to change how we think about research, how we do research, and the outcomes of it. So that's the Common Fund. The Common Fund Data Ecosystem, or CFDE, which we're representing today, fosters scientific discovery by promoting accessibility, use and reuse, and integration of multiple Common Fund programs in terms of both their data and their resources. And it does that by building a cohesive ecosystem. So the major structure is that you have these 5 centers, which are built to support the various CFDE programs, which you might hear called DCCs, or Data Coordinating Centers. The 5 centers that support CFDE enable users to query and use multiple Common Fund data sets. That's the Data Resource Center. They perform training and outreach, so that's our training center, which is responsible for the podcast that Mackenzie just told you about, among other things. The cloud computing, training, and support is provided by our Cloud Workspace Implementation Center, or QUIC, which is coming online to provide infrastructure so that you can do your CFD analyses in the cloud. Finally, you can derive knowledge from CFDE resources through our team, the Knowledge Center. And I'm going to talk more extensively about what we do today. All of these efforts are brought together by a centralized Integration and Coordination Center, or ICC, which keeps everything going. So that's a lot of information to tie it all together. Common Fund funds these Common Fund programs, or DCCs. A subset of those are integrated into the Common Fund data ecosystem so that individually and collaboratively through various formal and informal partnerships, those programs can transform research. And this work is supported by the action of our 5 centers. So recently, many of us were fortunate to attend a spring meeting for CFDE. We actually just got back from that this Wednesday. And I love attending these meetings every year because we learn some of the big picture themes, right? It's easy to get caught up in your own research and addressing your own questions. But if you can take a look at the broader ecosystem, you get an idea of where we should be headed next. These were some of the questions that came out of this meeting. How do we transform the tools that have been built within CFDE into what we call killer apps, right? These are the things that everybody knows the name of and everybody uses to catalyze their research. So the goal is not just to have various tools, but to have things that are really useful and used by the community. How do we make our tools useful for biologists? So not just computational folks, but also the folks doing wet bench work in the lab. How do we get users to innovate with CFDE data and tools? And finally, how do we connect CFDE to other external networks to broaden our reach? So these are things that we've been keeping in mind, and would love to work with the community to enhance your experience so that we can address some of these goals. So that brings us to specifics of our work within the CFDE Knowledge Center. So again, we're one of those 5 centers that support actions of the CFDE. And our goal is to improve understanding and visibility of CFDE resources. We generate knowledge by analyzing CFDE data and putting it together into a user-friendly and centralized entry point. And our work is designed to be both integrative and also focused around disease so that you can make relevant discoveries for your research. We do this by defining knowledge in various ways and by addressing several types of knowledge through the work that we do. So the first is foundational knowledge. So this is by defining the major purpose of every CFDE program. The second is collating primary knowledge. So those are published findings. Secondary knowledge, which can be obtained from a single CFDE program, but isn't necessarily something that they publish. And finally, we bring in tertiary knowledge. So these are assertions that are created integrating data from multiple CFDE programs. And this, as you can imagine, is the really exciting part of CFDE, right? It's to take resources generated by multiple programs, bring it together into something new and catalytic for discovery. And our ultimate goal, again, is to create something that's useful to the broader research community. So through the CFDE Knowledge Center, you can do 2 major things. You can learn about CFDE and you can analyze CFDE data. And I'm going to go through a bit of what you can do for each of those things. So to learn about CFDE, we have educational events like the webinar today and like some of the outreach activities that Mackenzie told you about earlier. But you can also do that on our website. So that's cfdeknowledge.org if you'd like to head over there and play around with it. If you click on any of our programs within the schematic on our landing page or up here on the top, you'll be taken to these sorts of pages. Now, these are curated foundational knowledge pages with some of the major take-homes from each CFDE program. This will give you an idea of what each program is about, what some of their major deliverables and biological questions are, and some of the key research that has come out of each program. You can also learn what resources have been generated by CFDE programs and how to access them. So again, through our landing page, you can click on, say, transcriptomics and learn all of the CFDE programs that have generated or are in the process of generating that type of data. If you go to those foundational knowledge pages that we just told you about, you can also learn about additional types of data that are not omics. So a great example of this is Spark, which studies the nervous system and neuromodulation pathways. And here you can see that Spark has some omics available, but many other rich sources of data that are not omic, but incredibly useful for the community. And outside of data, they also have other resources like methods, standards, and experimental and computational resources as well. So the second major thing, as I mentioned, that you can do through CFDE Knowledge Center is to analyze CFDE data. So I'll go through a couple of the tools that we have for you to do that. The first is to analyze genes or phenotypes. And this is one of the examples of secondary knowledge that we bring to the community. You can, from our homepage, type the name of your favorite gene and click here to go to the KC or Knowledge Center portal gene page. And that'll take you to a list of various types of summarized knowledge of that gene from not just one CFDE program, but from across many CFDE programs. And those are available exclusively here on the Knowledge Center website. The idea here, again, is to take information from multiple programs and put it all in one place. We have many examples of this, so I encourage you to play around and see what you can learn there. One example is integrated multi-tissue single cell maps. So these are available from HubMap, but you can see interactive view maps, in this case of the gene BDH2 in various heart cells. It's interactive, so you can toggle on and off to look at different cell types. And you can see specialized visualizations like these violin plots of gene expression within each cell type. You can also get things like examples from human genetics from rare disease cohorts. This is a cohort from Gabriella Miller Kids First, or Kids First program within CFDE. Kids First studies various rare pediatric structural birth defects and rare pediatric cancers. And the ultimate outcome on the Knowledge Center is this sort of view of variants and their consequences for all phenotypes within the Kids First cohort. So here you can see each variant that's present within the Kids First cohort, its frequency within NOMAD population databases, and then you can click on the View Evidence button to get a bird's eye view of the types of rare phenotypes that have information for this variant. I'll give you an example of a biological finding that's come out of this module. So this paper was published a couple years ago, and this performed whole genome sequencing for a cohort of individuals with non-syndromic cleft lip and palate. If you search for these genes, you can actually find these particular variants that were reported here in the paper within the Kids First table. And you can easily see that these variants are present within the Kids First cohort, but absent from the NOMAD population database. You can also search for phenotypes in the same way as you search for genes. So an example of a finding that you can get here is provided by illuminating the druggable genome, or IDG. This is another CFDE program that studies understudied drug targets of various sorts. And here you can see potential drug targets for various phenotypes provided by that program.
Julie, Eve has a question, so I'm gonna allow Eva to ask it. I just need to, okay, here we go.
Great.
Okay, I think we can ask a quick question and just pause there.
Sounds good.
What I did was I put it into the questions, I think, if you can see it. Sometimes when we have chat and um,
Oh,
questions both open, can you see it? So a
Oh,
man actually asked this on Blue Sky this morning. A physician, a PhD research person, was
mm-hmm.
looking at gestational diabetes, and I thought about, or not gestational, but a problem that causes it's hypertension in diabetes, and it's an interaction between the kidneys and several other problems. Now, if he were going to use your portal,
Mm-hmm.
he might need to tell you the phenotypes that women might have, like gestational diabetes. Now she's got hypertension, and we have diagnosed her with preeclampsia. Is it possible for someone at Broad Institute to query it like that? That we don't need to get involved in doing it right now with the portal, but is it possible?
So let me
Think
just make sure that I'm understanding your question so that I can tell you what you could do with the tools that we've developed. I guess our tools are not as centered around diagnostics per se, um, but what we could help somebody to do, somebody who's more in the basic research space, is if they had, say, a phenotype, so let's say preeclampsia or hypertension or some specific phenotype name, they could run that through that module that I just told you about and see if there are predicted drug targets or expression profiles or other information of that nature for that gene. And then the other thing that they could do with that actually ties in nicely to our next part of the talk that we're going to get into. I do want to emphasize that it's not as clinically focused. This is more meant to fuel sort of more basic science research at this point.
Oh, no, that's perfect. I actually asked the question so we could kind of get there. I mean, because think I know how to get at the basic, the pathophys part from the data I've pulled over the last 9 years. And then
Mhm.
we might query you and see what you thought, as far as genes and whether there's particular populations that are known to have the problem. Does that,
Sure.
thank you so much for answering. You can mute me, please.
Yeah, no problem. Thank you. That's a great
Thank you,
question.
guys.
Yeah. All right. Well, great. Um, so with that, we're going to get into the next part. Actually, Eva, you set us up really nicely for the next part of the talk, where we're going to talk about another tool that we've developed, again, in the more basic science research space. But the goal of this tool is to connect gene sets to human phenotypes. And some of you might have heard about this before, but we've recently expanded the functionality of this tool. So I'm going to go into it a little bit more. So to take a step back, what are we looking at here? We're looking at gene sets. So as the name suggests, these are just sets of genes, right? And these can be generated by many experiments. One example would be something like a differential expression analysis to look at genes that are up or down regulated in a given disease versus controls. Because this is a relatively simple sort of output, it can come from many types of experiments and be generated by many types of CFDE programs. So it's sort of a means of connecting all these disparate data types across the ecosystem. These are also generated, excuse me, by external sources. So in that way, it's a means to connect to other ecosystems outside of CFDE as well, and to broaden our reach. And finally, gene sets are powerful because they enable us to move beyond looking at just an individual gene and its role in biology, but to expand to look at pathways and mechanisms. And we'll go into ways that we've come up with to do that. So these are simple, they're concrete, they're easy to generate. As of the spring meeting, we actually learned that there are now upwards of 1 million gene sets that have been identified from our colleagues at the Data Resource Center in CFDE, which is fantastic. So they're packed with information, but they're simple enough to be able to be linked to one another and to provide quantitative and qualitative data in the ecosystem. So questions that are addressed specifically by our knowledge center gene set analyses are which gene sets capture the important mechanisms and which mechanisms are actually relevant to human health. We do that using a method developed by Jason Flanick from the Knowledge Center, and it's called PIGEAN, Priors Inferred from Gene Annotation. So PIGEAN is a Bayesian method that jointly models the probability that each gene is involved in a phenotype. It does that using two major pieces of information. The first is genome-wide association study, genetic association statistics. So these are provided for variants that are near to the gene. And we call that one line of direct evidence for involvement in a trait. PIGEAN also takes into account the gene sets that contain the gene. So these are not a form of direct evidence, but they're indirect evidence of involvement in a trait. And using these two pieces of information, PIGEAN is able to connect gene sets derived from across CFDE programs and from intersections from among the CFDE programs and connect them to various human phenotypes, additional genes, and mechanisms. The major readouts of PIGEAN results are for each gene set, estimates of the effect of that gene set on the probability that those genes within the gene set are involved in the disease. So these estimates are available both for the gene set in isolation. So we call that the marginal effect. That's available here through the results table. Or the results within a joint model. So that considers not only the gene set that you're looking at, but all other gene sets that you've tested. So it provides a sort of testing correction. We call that the joint effect. That's available here. In addition, for each gene, PIGEAN results give you estimates of the probability that that particular gene is involved in the disease. So again, the estimates are provided in these three major formats. The first is this combined genetic support score, the direct genetic support score, which contains only the GWAS association measurements, And finally, the indirect genetic support score. So that's the score provided only from the gene sets that the gene is in. One example of a finding that you can get from this type of analysis is from one of our CFDE programs, MotorPack, which studies the molecular effects of exercise. In this case, you can see that the readout is a phenotype of reticulocyte count, so those are red blood cells. And you can see that that phenotype is connected to this gene set here. So this is genes that at time point 30, after 2 weeks of exercise, are upregulated in the blood of male rats. If you expand to see the genes in the gene set, you can see all the genes that either directly or indirectly contributed to the support of this connection between this phenotype and the gene set. So what you can infer from these results are a few things. First of all, that this gene set is important, right? So this is a biologically interesting finding. The genes within this gene set have roles in the reticulocyte count phenotype. So that's the connection to the human trait within that table. You can see which specific genes are involved in mediating this genetic effect. And finally, you can see causality. So in this case, we know that there are genes that have a causal role in this phenotype. So this is a really powerful readout. The table itself is packed with information, but if you break it down a little bit, you can get a lot of valuable gems for your research. So we presented a basic version of PIGEAN back in October with our last CFDE meeting, but now we've added some exciting new additions. So we've updated the PIGEAN module, so we now have nearly 7,000 total traits. These are from our Association to Function Knowledge portal, which studies various common disease phenotypes through genome-wide association studies. We've also integrated additional GWAS information from the GWAS catalog through our collaborators at EBI. And finally, we've integrated over 1,500 rare disease traits, and these are from Orphanet. Another expansion that we've made is a new feature, and this is in beta, so we're still very much expanding the functionality of this feature. But we wanted to tell you about it so that we can begin testing it and improving this interface to make it more useful for people's research. So this is a feature that actually allows you to find other genes that are predicted to be in your input gene list. We call it BY Bioggle, bring your own gene list. And you can access it from this KC Labs tab here at the top of our homepage. This allows you to search for a set of genes, either separated by line or by semicolon like this. And it can enable you to find some really interesting things. So this is an extension of what I showed you earlier with the kids' first result from our secondary analyses on the KC site. I showed you that on our Knowledge Center website, you can obtain a list of genes that have been previously reported to be associated with human rare development traits, including cleft palate. So I mentioned these 4 genes that had been previously reported in association with cleft palate were found in our Kids First module. If you take those 4 genes and input them into this Bioggle interface, hit search, you get an output that looks like this. Now, these are genes that are predicted to be in your gene list. The striking thing here is that if you query each of these genes in Online Mendelian Inheritance in Man, a forum for rare disease research, you find that actually 8 of these 10 genes have also been previously reported in association with cleft palate. The other 2 are not overtly associated with human cleft palate, but I can tell you that IHH in particular has been associated with cleft palate in mouse. And all of these genes are in the same sort of sonic hedgehog signaling pathway. So you can imagine that over time, a module like this could help you predict additional strong candidate genes for a trait of interest based on their involvement in your pathway. So over time, we hope to use these modules that we've developed to expand gene set analyses and make an impact within CFDE. So our ultimate goal is to curate, standardize, and integrate new and existing CFDE-generated gene sets, to streamline the user experience and promote usage and utility of these gene set tools. We want users to be able to analyze their own gene sets and synthesize their results into interesting scientific findings, both in CFDE and beyond. We're working with the CFDE programs to return some of the interesting findings to them and to validate some of them through validation challenges. So that's something that we're in the process of getting going within the Broad. And finally, we want to extend to new target communities. So that's where all of you come in. It's hard to know who might want to use your tool or what types of uses they could conceive for your tool until you talk to people. So we're really excited to hear what you all think and if there are ways that we could expand these interfaces to make them useful to you. So finally, I'm going to talk about one additional beta feature that we've developed with this recent release. So the goal here is to reach out to a new community beyond our sort of, bread and butter human genetics community, and that's the mouse community. The goal of this module is to connect mouse and human phenotypes. So the rationale for developing this module comes from a well-known problem, which is that preclinical trials are often done in animal models for obvious reasons, but the findings from animal models don't always translate to success in humans in clinical trials. Now, some types of models are more successful than others, and some types of diseases are more successful than others. But often the problem is that the phenotypic similarity itself does not imply genetic similarity. So it's hard to know in which cases you should trust the results most. So most existing sites for mouse research are incredibly powerful for looking at semantic similarity. So if you want to find, a gene, you can type it in here and see what the mouse ortholog of that gene, what phenotypes it's actually involved in. And if, say, you're interested in a human gene that's been associated with hearing impairment, you can look and see if that mouse also has hearing impairment. The new innovation that comes with what we're presenting to you today is the idea that you can actually leverage PIGEAN, the method that I just told you about, to connect mouse and human phenotypes. So this is different because instead of matching phenotypes based on semantic similarity, so looking at head bobbing in mouse versus head bobbing in humans, you are looking at genetic similarity to find directly genetically supported connections. So the way that this module works is by inputting gene sets that are specifically related to mouse phenotypes. Those come from IMPC, MGI, and MSigDB. We then connect those again to the common and rare disease human phenotypes as before, and use PIGEAN to run those mouse-specific gene sets through PIGEAN. Now, because those mouse gene sets are related to a specific mouse phenotype, we're able to use those as a proxy for connecting that mouse phenotype to that human phenotype. So the end result is that you find genetically associated phenotypes from the opposite species in each case. We have two major interfaces at this point. The first allows you to query by a mouse phenotype. The output is the top 20 human phenotypes that are genetically linked to that queried phenotype. So in this case, we put in head bobbing as our phenotype, and we see that human rare deafness is the top human phenotype that's linked to it. Again, this is through genetic similarity so we can actually trust that this association is strong. Similarly, you can query by a human phenotype. So in this case, the result is the top mouse phenotypes that are genetically related to that human trait. I'll give you an example of a result that could come out of this type of a search. If you want to search for a human phenotype, you can put it in the search box here. In this case, I searched for a rare disorder of human eye movement. It's called congenital fibrosis of the extraocular muscles. And the results actually come up with some interesting things, right? You see some things that you would expect here, like abnormal extraocular muscle morphology or abnormalities of the cranial nerves that are involved in moving the eyes. But you also find some things that are unexpected. For instance, abnormal basal ganglion morphology. So the basal ganglion is another part of the brain that is not directly involved in moving the eyes. If you study these human disorders, you know that this brain malformation can actually also be seen in many forms of the human disorder. Experienced neuroradiologists in human research know to assess for it, but other people might not. So you can imagine for lesser known sort of phenotypic associations, it might be of interest to see what other phenotypes have been seen in a mouse that could be related to the human phenotype that you put in. So this is one example of the power of this tool, that it can pull out sort of unexpected phenotypic connections that might not have come to the tip of the tongue. So for next steps, we're working on beefing up this user interface and how we organize the data to make it more accessible to our users. We want to validate it and compare it to some of the existing approaches, especially those tools that have been developed for semantic similarity, to see how it compares and what the advantages and limitations are. And finally, we want to know what our mouse geneticist friend thinks. We all can have many different uses for this. I've just given an example of how I might approach it as a human researcher, but we really want to hear what you think as well. All right, so with that, thank you very much. We really want to hear from you moving forward. So if there are types of tools or research resources that you want to apply to your studies, let us know. If there are trainings that would help you, we work closely with the training center, so we can let them know and they can work on developing those out. If there are public resources that you use regularly for your research and that we can tie in with some of the tools that we've developed here, we'd love to hear from you too. So with that, I'd like to thank all of my colleagues at the Knowledge Center. We really have a wonderful team that worked very, very hard to make these updates. And I'm really excited that we can share it with you today. So with that, thank you very much. And I will take any questions. See, Eva?
I did put it also in I think the chat, but basically this is awesome. We've come a long way and we're going to go forward. I think what's going to need to happen is at university settings, physicians are dealing with clinical problems. They're busy, they're overworked. We're going to have to step back a little bit and say, okay, this is what we see in clinical care. This is what it looks like. And we know it has this and this, lab problem. The lab work is going to look this way. And we know the signs and symptoms are this way. And then we will be able to look at the mouse model once you bounce off the similarity and differences and know whether we're really looking at a human genetic problem or we're looking at something in the environment that's inducing like hypertension or obesity. And so, yeah, this is so great. I just want to thank you so much for it because I think I heard a portion of this web presentation before, but I had it was a big windstorm and I had internet problems. So, but thank you guys so much. It's awesome your progress.
Well, no,
Thank
thank you, Eva. And we appreciate hearing from you as somebody that's been in the clinical realm. to know how we can think about connecting there too. So thank you for that.
Okay, we
Thank
have
you.
some other questions in the question and answer. And so one of them is, thank you for an interesting presentation. You've demonstrated how to analyze a single gene phenotype, and I am wondering, how can we check results for a list of genes? Does the website support some form of programmatic approach, such as a reusable code for this?
So we're working on developing out APIs for a lot of this. That work is very much in progress. So the example that I showed you of the new beta tool that allows you to put in a list of genes and to come out with your own results is one example of how we're doing that. But this is still very much something that we're in the process of developing. So I would say stay tuned for that. That is one of our ultimate goals here.
Did you mean you wanted to run it through PIGEAN, or I didn't fully hear the question, Julie? Was the question, because I was going to mention the Terra workspace. Does that apply to this?
Oh, I see. Yeah. So that I guess within the Terra workspace, we've also primarily applied it to use for PIGEAN results as well. I'm not sure who asked that question, if they want to elaborate a little bit more.
You want to raise your hand so we can unmute you, or I'll try and look at the question too, or maybe we can.
Oh, yeah.
Hi.
Go ahead.
Hi, can you hear me?
Yeah, we can hear you. Hi.
Yeah, okay. So, my question is, like you mentioned that you are opening an API for future use. So I think it's very good ways to do a reusable, and like you can do it over and over again. Sometimes at exploratory stage of research, you have a gene set or a couple of them, and then you want to check. So that's all my question, and I'll definitely looking forward to the future to see development of the tool. Thank you. Okay.
Sure. Thank you. That agree. That's something that can really help to I guess fit in with the broader mission of CFDE as well, right? Which is to promote fairness and general reusage of the data. So yes, that's something we're actively working on. Thank you.
Okay, there's another question in question and answer. I use DisGNet, hopefully I'm saying that right, for my computer-aided target identification. Will CFDE play some role in replacing such data platforms by offering open source integration of multi-omics data for human phenotype?
Let's see. So I'm not as familiar with that particular platform, so I can't speak to that specifically. Or is this for drug target identification?
I'm not certain who asked the question, otherwise I would allow them to talk. So I guess if the person who submitted this can raise their hand, I can give you that permission to talk.
I guess in the meantime, while we're waiting, I can say that the IDG that I mentioned, the Illuminating the Druggable Genome, their focus has been on looking at historically understudied sort of like, dark proteins that have the high potential to be druggable. So these fall within classes like GPCRs, ion channels, and kinases that have historically modeled quite well in drug studies. And IDG has aimed to compile various types of research on those understudied drug classes to determine which are most likely to be useful for potential drug applications. So I
Good
can
point,
speak to
Julie.
that. Yeah.
One part of that, I think if I understand your question while you're unmuting, is I think that ideally, yes, CFD being open access, absolutely. Our goal is to make it so targets are, you could use this for what I would call genetic support, and this and other resources that we've developed in our group, because you have a particular phenotype that you study. Will give you, through the module that Julie went through today, you get a list of genes that are most likely connected to that phenotype. And you also, over time, will actually get like a multifactorial breakdown of different pathways associated with that. That's going to come online actually in the next couple months. It's based on this PIGEAN method. It's in beta testing right now. So if you're actually really curious to see that, we'd actually love to talk to you about what you would need and what you find very valuable about the resource that you use, that we can make sure that we like bake into the presentation of these results for any genetic target for common and rare disease. So we'd love to talk to you.
Sorry, I'm still uncertain who the person asked the question was, but they confirmed in the question and answer that it is for drug target identification.
Great, thank you. And just to follow up on what Noelle said, if you do want to reach out to us, go to our Knowledge Center website, the cfdeknowledge.org, and we have a help desk email. So if you want to follow up with us and give us more specifics as to the type of application you'd be looking for, we can follow up with you there. Thank you.
Any
Any
other
other
questions? Yeah, sorry, Julie, the same thought. Okay, again, if you have any other questions that you weren't able to ask today, like Julie said, please feel free to reach out to us. And thank you again for attending today's session of the webinar series. As a reminder, our next one will be on April 25th, and we look forward to seeing you all then. So thank you, everyone.